Hello. This is a short video to look at a very important tool that's also a website. So you can go to it from anywhere, anytime you want. So this is ViralZone. ViralZone.xpazy.org is the way you get there. Or you just type in ViralZone in your browser and then click on the first link. That should get you there. What they've recently done, and this is the late 2024 version, so the future is unknowable, and they update this thing all the time. It will look different probably when you get here, just because that's the way it is. And just give a moment of appreciation for those little crazy little Ronas up there running around. Love it. Yeah. So what they've done now is they've actually made this match up with viral taxonomy, and that is a good thing. So in each of these groups, they're actually, when you mouse over it, they're going to tell you what these groups have in common. And the buttons that used to be buttons at the top aren't there anymore, but all these other things are. And so you can see, like, okay, double-stranded DNA, these are all dsDNA things. And you can look inside, and they will tell you what these all have in common. So we'll have an HK97-like capsid protein that's in common to everything in this group. And everything over here will also have HK97 capsid. And they're going to be they're going to have different morphologies depending on the tail. Those aren't taxonomy. Those are how they look. It's like you say sports car, but it doesn't tell you which factory it came from. It's just like a general type of thing, general class of things. And over here, so when we see something like cell type capping, you can go over it, but cell type capping is going to involve an RNA triphosphatase. It's going to involve a guanylyl transferase, because it's usually going to be GDP that's going to be added, or we're going to add it to a nucleotide where we've taken off one of the phosphates. Guanylyl transferase is going to put on the backwards G nucleotide, and then we're going to have two different methylation steps, which are going to be the N7 and the 2' O methylation. This is all stuff you learned about in the coronavirus chapter, I would assume. And so, everything in this group appears to share those common traits. And so, all right, those are all the double-stranded DNA viruses. And so they include things like herpes viruses. You like herpes, don't you? There they are, yeah. Also these other crazy viruses that are over here. These are mostly bacteriophage that are over here, but there's so many different kinds of bacteriophage. Even throwing that word around, it basically means nothing, because there are bacteriophage that are RNA only, DNA only, enveloped, not enveloped. Anything that one is, another of them isn't, and so you really want an entire course on bacteriophage. That's what I think you want. Here's our adenoviria. These are the crazy ones that are going to infect. They're going to infect those different extremophile archaeans. There we go. Archaeal filamentous viruses, and they're going to have that weird A-form DNA, and so it's going to look like this. So it's not B-form DNA and it's not Z-form DNA. And they're going to have characteristic major capsid proteins. And then if we go to some of the other groups down here, like Viridinaviria, these are some of the weirdos. So these are going to have also shared characteristics. So they'll be, these are single-stranded DNA, these are double-stranded DNA. But they're going to have a DNA polymerase, and they won't cap any of their own RNAs or something like that. Which means they have some other mechanism that's able to do that. These are going to cap their own RNAs, and they're going to cap them in a way that makes it look just like cellular RNAs, probably because they're trying to hide inside of a cell with normal eukaryotic cell RNAs. And so this is like African swine fever virus. Pox viruses are down here in this group. These are the Phycodina viruses, those great big sort of plant viruses that almost look like a cell lost a couple too many components and it can't even be a parasite anymore. It's now just doomed to be a virus and thrive forever. Yeah, poor thing. And a whole bunch of other weirdos. Yeah, look at them all. Look at them all. And then you got your unassigned things over here. Oops, sorry. Let's stick on that. And so these are going to include baculoviruses, which we put up there. Nudie-viruses are kind of neat. And if you go into these, they should always still have little little captions. And so, yeah, little circular double-stranded DNA that makes a nice little helical capsid. Has a nice little membrane protein that's going to act like a little spike. There you go. So you can see what they do. You can see some three-dimensional models from viruses like this. And yeah, in general, you learn to understand your virus world. So rather than go through all of these things, let's just take this to one of my favorite groups of viruses. Let's look at the positive stranded viruses, which are Pisu viricota. Well, actually, before that, let's look at mono DNA, so single-stranded DNA. Now, these guys are neat. So most of them have not a shared protein, but a shared way of copying their protein. So replication initiation endonuclease. So they're going to do rolling circle replication where they go round and round and round. And so that means they're going to make a little nick in it before they can actually go around. And so they're going to actually encode their own gene to do the nicking for the most part. Although there are some cellular genes that will also do that job, and some other viruses will use that. And so, yeah, these have a polymerase or a replicase, or they don't, yeah. And they don't have a capsid gene, but they do have an envelope. These pleolipoviruses are actually, yeah, enveloped viruses, enveloped single-stranded DNA viruses, but of bacteria. How do they get in? How do you make that work when you're infecting something with a cell wall? I certainly am not the one to tell you, but they do work very well. That's for sure. Here's our Gemini viruses that make the sort of double capsid. They're kind of neat. Let's look at those. There you go. That's a Gemini virus type capsid. Two regular capsids, but the genome got a little too big for its britches, and so they had to diversify and make this sort of double capsid monstrosity. It's fine. They're easier to recognize, and those also get stolen by some of the other viruses. And so on. Yeah, enoviruses, I believe those are the ones that make the cholera toxin phage. And there's a bunch of other crazy things that are in there. Let's go over here to positive stranded RNA viruses. And those are going to include our very favorite ones, which are things like nidoviruses. And so you can see that these are going to be in the same group, Pisoni viricites. And so these are going to have Sobelli viruses and Picorna viruses and Nido. So Pisoni viricites is the ending. So there you go. And so what do these have in common? Well, they're going to have an RNA-dependent RNA polymerase in the PFAM1 family. So that's just a particular type that is common to all of these things, and they do have this. These guys are both going to have a VPG, and now you actually know enough to appreciate that. And you can say, oh, VPG, that's the protein primer for RNA synthesis. And it is, yeah. And so they're going to use that in place of a cap. And then you got your nidoviruses over here that don't have a VPG, but they sure have something that works the same way. It's just they will then take it off and put a normal cap on it. And so they'll do capping with the enzyme, the NERAN enzyme, and then adding the right methyl groups with their methyl transferases. Yeah. And so on. You got all your other crazy things here. Hypoviruses are over here. These are the ones that seem to be just naked RNA that is able to spread within fungi. And yeah, lots of other cool things. You got all your negative viruses, your circular single-stranded RNA things, which are going to include basically viroids, and also some of these other groups. Pretty please. There we go. Yeah. And so some of your viroids will be in here, and they're listed as not viruses, and darned if they aren't on ViralZone, listed amongst the viruses. There are, things that are like, so viroids are like one of these things, these little RNA viruses, RNA viroid-like things that do encode a protein. But they still don't encode enough to really truly be viruses in my book, but they're kind of categorized as if they were. And you got some of these crazy guys over here. And I think I know which one we're looking at. Wait for the picture to come in. Wait for it. Oh, no. Picture's not coming in. Oh, well. Yeah. So, not encoding proteins, but or actually no, these would sometimes have both proteins and ribozymes. So it's like a RNA virus and a ribozyme had a love child. Yeah, I don't know why they would love each other, but why not? Yeah, they're both terrible. Maybe that's what attracts viruses and people to each other. Who can tell? So, ViralZone is now an easier way to look at the ICTV classification, and while ViralZone lags behind ICTV, ICTV is mostly just names and some pictures if you really dig down in deep. And ViralZone, I think, is a lot more friendly and accessible, but they're both coming from the same place, which is to show you the world of viruses and all the crazy stuff that's out there. And they are both doing their best to keep up with the insane new trends in types of viruses and not quite viruses and really not a viruses, but whatever. Put them on the same page kinds of things. And so, yeah, that's what you have over in this entire group. And this is one of the realms of the viruses, even though they're not. So that's enough, I think. These things are maybe not all related to each other necessarily, but they are close enough to each other and they have a couple of common parts that it looks as though they may have at least stolen from the same source, and they may be doing the same sorts of things. And maybe that's as close as you can get. Humans love to categorize things, and we have tried our best. And maybe in the future, you will see a totally sensible system that fits all these things in seamlessly, unified theory of everything. I doubt it, though. Yeah. And even if we do come up with that, viruses will find some way to break it. I think you can sleep easy banking on that. So thank you very much.